Hey everybody, John Marshall back from the Ruesch Center 2018 Fighting a Smarter War Against Cancer, and I'm joined with three very incredibly smart and innovative folks who are helping to drive our science of precision medicine, a rapidly evolving field both on the technology side, how to measure, how best to measure, as well as the therapeutic side. So absolutely embedded into our day-to-day practice, so you need to stop whatever you're doing, put your phone down, and listen to what these guys are about to say. John Strickler, I want to start with you. Thanks for coming up, by the way, from the great state of North Carolina.
Appreciate the invitation.
Go Devils. Give us a little sense of what you've shared with us today and sort of how this is playing out as the clinician on the panel in day-to-day.
Yeah, so essentially what's going on in oncology is increasingly we are learning about the biomarkers that drive resistance to the therapies that we give. And increasingly we're finding that a simple tumor tissue biopsy does not always tell us everything we need to know about that patient's tumor in terms of how to target that tumor, how to bring that patient the best treatments possible. So what I presented was some data around this idea of EGFR antibody rechallenge, and essentially using what's called a liquid biopsy, where we can sample the blood of a patient and analyze tumor DNA from a routine blood draw to identify those patients who might benefit from rechallenge with an EGFR antibody.
And so what's that mean? So I'm used to getting tissue, I'm getting a RAS mutation or not, right or left.
Yeah.
I'm deciding on EGFR therapy. I give it, maybe for a while, it works or it doesn't work. So you're saying then do a biopsy like they do in lung cancer, a liquid biopsy, and measure for a specific resistance pathway?
Yeah, I mean, that's exactly right. So what we found is that tumors are pretty darn smart, right? They respond to selective pressure. When we give them a treatment, they have to try to figure out a way to outsmart that treatment. And so when we get that initial biopsy, we learn a lot about the tumor, but then we give the patient a bunch of therapies that then change that tumor, force that tumor to evolve in a way that allows it to escape the treatment. And that's where the liquid biopsy comes in, as a way to sample what happened to that tumor that made an initially sensitive tumor suddenly resistant.
And so when you find this marker or whatever, does that then enable you to target with new therapies or new combinations?
That's absolutely right. So we are looking for a pattern of resistance in patients that would either tell us, yes, we can rechallenge that patient with a therapy, or no, we shouldn't rechallenge that patient with a therapy. Yeah. And what's nice for the patient is that instead of having a biopsy of a liver or a lung or something else which has cost, which has risk, we can do all of this off of a simple blood draw.
They love having biopsies. What are you talking about? Well, thank you. It was really exciting, and your talk was of course, fabulous. Sitting next to you, David Spetzler, Chief Scientific Officer for Caris. Tissue-based primarily.
Yes.
Talk a little bit about not only where we are today with sort of tissue-based testing, but some of the thoughts that you're going to share with us today about where it's going.
Yeah, so again, thank you for the invitation. Where we're focused on is trying to escalate what it means to measure biomarkers. So right now there's a pretty simplistic paradigm where we're looking for generally one biomarker associating with one drug. And that really is underwhelming compared to the inherent complexity of the tumor environment and how patients are responding to various therapeutic regimens. It's pretty rare nowadays to give just a single monotherapy. So what we're looking at is combining DNA measurements with RNA measurements with protein measurements and using sophisticated algorithms to determine how patients are more likely to respond to specific regimens. And so today, actually, we're going to be showing for the first time data using a predictive signature to determine which patients should be going towards full FOX versus full Fury in that frontline metastatic colorectal cancer setting.
So this is like Groundhog Day. What are you telling me? You're going to tell me which chemo to use again? We've been there with you all, Caris, and other companies. So do you think we've got new evidence that would support giving me better insights as to which chemos to use?
Yeah, absolutely. I think that one of the key evolutions of biomarker testing is really beginning to assimilate massive amounts of data. So the tumors are smart, and so looking at just one marker or two markers really is not going to be able to decipher the complexity of what's going on, especially as you start to consider the fact that there are interactions occurring within the drug regimens that you're dealing with. And so when we start to look at the signals and where they're being derived from, it's combinations of hundreds of different molecular features that tell you whether or not a particular patient is going to be more sensitive or less sensitive to these broad-based chemo regimens. And so that's what we're going to be talking about.
Tissue-based therapy, I mean, a lot of people are talking about liquid biopsies. There's a lot of talk about this and a lot of excitement. From Garden Health,
Yes.
Leslie Kiedrowski
Yes.
is joining us today to talk about this really cool concept of the liquid biopsy. Clearly in the marketplace, people are ordering them. We like this idea. I always use this analogy that in the blood is sort of the garbage, everything that's out there, so whatever is there would be picked up there. Mm-hmm. Give us a little background and update on what you're going to talk about today around liquid biopsy as a way to measure for precision medicine.
Absolutely. Thank you so much for having me. And what I plan to discuss today really builds on what John has already mentioned, which is this idea that tumors are shedding bits of their DNA into the bloodstream, and we can use that as a tool to pick up what's going on in a patient's cancer at a given moment in terms of the genomic profile. And one of the great strengths of cell-free DNA and this liquid biopsy idea is we can gather not just what's going on in one area of a lesion that we can hit with a needle and test, but can really capture that whole picture of global heterogeneity across a patient's burden of disease. I think that's really useful in some cancers for doing treatment selection when there's heterogeneity at the start, like gastroesophageal cancer, which I'll be discussing, but also especially important in the setting of acquired resistance, where we often see multiple subclonal resistance mechanisms popping up that can be captured really nicely with cell-free DNA.
Yeah, you guys have got a really strong role when I look at this field in lung cancer, for sure. Looking at emergence of mutations. We've got drugs to target. But there's also the concept that you know what you're looking for. There's that sort of hunt. Could you talk a little bit about using liquid biopsies as the sort of screening tool versus one looking for very specific emergence of resistance?
Absolutely. Do you mean screening in earlier stage
Yeah,
disease?
just I got a new patient. They're sitting there, and I want to know their molecular profile.
Yep, absolutely.
So I've got different people knocking on my door saying, "Use my test. Use my test." Liquid seems better. Patients are aware of this. Can you talk a little bit about that as
Sure.
a front line versus a clear mark indication that you've got now for following patients?
Absolutely. So in the front line, I think there's definitely a push in a lot of areas for more comprehensive genomic profiling, not just zooming in on one marker or another. And blood is certainly a patient-friendly option. As you know, I know they love the biopsies, but
They
a
do
blood draw
love the
is-
biopsies.
blood
You're
draw is
taking
much
tumor
easier.
away from them.
Yes, they don't want that tumor anymore. But I think the idea is using something like a next-generation sequencing panel or doing that comprehensive approach can pick up on treatment targets sometimes that maybe are not expected or anticipated. Things like MSI or now NTRK fusions, for example, that do have approvals across all cancers. They may be rare, but we can pick them up even if we weren't necessarily going for that one specific thing. And that can change what's going on in the front line. And there are also a lot of really promising targetable agents looking at gene amplifications and things like that, that maybe a patient would be eligible for a clinical trial for a drug that will be effective. Not to mention, you also get a picture up front of what that tumor looks like at baseline, which can really help to interpret those results that you get later on if the patient is progressing. And as Dr. Strickler said, that tumor's continuing to evolve if you know what it started like.
Yeah, John, I struggle with this a lot. I'm good at interpreting, say, a CAT scan. I know its quality. I know how it was done. I know if it needs to be. I know if it answers my question, right? And I know if I need to order a PET or an MRI, right? I understand that I don't know how to do one, but I understand how to interpret it. With genetic testing, molecular testing, honestly, even though I've been in the space a while, I don't really understand it very well. And I get a report back, and I'm not sure what I'm looking at. I'm not sure of the quality. I'm not sure if it's black and white. Do I need to repeat it? This kind of thing. Can you talk a little bit about that evolution as clinicians and the reporting of these products?
Yeah, I mean, I agree with you. It can be very complex to look at a report and understand, okay, this is what I should do for my patient next. And there are a lot of reasons for that complexity. There's nuance. We're accustomed to thinking about these molecular markers as black and white, binary. KRAS mutation, yes, no, right? NRAS mutation, yes, no. But in truth, whether it's tissue or blood, there's some nuance to this and some subtleties. It could be the case that a certain percentage of those cells are KRAS mutated or NRAS mutated, or it could be that all of them have that mutation. And what you see in the blood report is a lot of that nuance on that sheet of paper, right? You'll see not just what mutations they have, but the frequency of the mutation. We're not as accustomed to seeing that nuance with the tumor tissue reports, but that may be something that comes in the future. And I think what that speaks to is a need for prospective validation of the assays in the context of future clinical trials. And I think that's where we're all headed over the next few years. And I think as we start to become more accustomed to reading these reports and then incorporating that data into prospective trials, all of us as clinicians will get more comfortable about making treatment decisions based on those reports.
We'll get better at it.
Yeah.
So thank you guys for joining. I think we can all agree, there's no question, we are in the same church around the importance of broad molecular profiling for our patients, particularly with metastatic disease, but maybe even more, depending on the disease. Clearly an evolving field with new technologies emerging and new approaches going forward, and docs need to keep up with this and know what they're looking at when the time comes. So again, thanks very much for all of this.
Thank you.
All right, thank you.
Thank you.